fourth step of aerobic respiration, which is called electron transport system. Sometimes it is called electron transport chain, followed by the so-called oxidative phosphorylation. So, the last and final event of complete oxidation of glucose molecule is the ETS followed by oxidative phosphorylation. So, where this event going to be occur, what are the important reactions to be occur and the result of this reaction. So, coming to this one, in sequence, it is the fourth and final step in aerobic respiration. It is not going to be seen during anaerobic respiration. And coming to the location where it occurs, the ETS and oxidative phosphorylation going to take place in inner mitochondrial membrane. So, the site of the fourth and final step of aerobic respiration, inner mitochondrial membrane. And you know the mitochondria has two unit membranes, outer membrane and inner membrane. And a very important feature of inner membrane of mitochondria is folded nature. So, this highly folded inner membrane of mitochondria with finger-like projections called cristae is the main site for completion of this ultimate step. So, inner mitochondrial membrane, or you can consider as a cristae, and this is the process which involves three important events. So, let us see what are those three events. During this process, the reduced coenzymes, whatever the reduced coenzymes that are formed before this reaction, like the NADH plus H pluses and FADH2s. These are called reduced coenzymes, and these are formed during glycolysis, that is already seen at the reaction where 3-phosphoglyceraldehyde is converting into 1,3-bisphosphoglyceraldehyde. There is an NADH. During link reaction, there are 2 NADHs. That Krebs cycle, there are 6 NADHs altogether formed. Along with FADH2s are formed, so they are called reduced coenzymes. They hold the electrons and the protons of glucose. They are supposed to be transfer the energetic electrons of glucose to the ultimate electron acceptor in order to generate ATP. So, these reduced coenzymes need to be oxidized. So, oxidation of reduced coenzymes is a first event, and this oxidation of reduced coenzymes is followed by reduction of atmospheric oxygen. So, naturally, here NADH and H pluses gain the electrons and protons from glucose during various steps of aerobic respiration. They are going to be faithfully transfer those energetic electrons of glucose to an ultimate electron acceptor. In this story, the ultimate electron acceptor happens to be one of the atmospheric gas, that is oxygen. If NADHs are oxidized, automatically the electron acceptor going to be reduced. So, reduction of oxygen into water. If oxygen accepts electrons and protons, it is a common chemical equation, H plus, 2 H pluses and 2 electrons and half O2 gives rise water. So, electrons from NADHs and protons from NADHs hand over to half molecule of oxygen to form 1 molecule of water. So, reduction of oxygen into water is another event in ETS. Finally, the energetic electrons transfer from NADH to oxygen help in the production or development of a proton gradient. So, there is a coupling of proton translocation with electron transfer. We will discuss in detail later. Because of that proton gradient creation, there is a possibility of synthesis of ATP from ADP and inorganic phosphate. So, the last step is synthesis of ATP. So, oxidation of reduced coenzymes, reduction of oxygen into water and synthesis of ATP. We can call it as oxidative phosphorylation. Oxidative phosphorylation. That means the ADP and inorganic phosphates are reacts and forms ATP by using an energy that is derived during the oxidation of NADHs and FADH2. So, during the oxidation of NADHs and FADH2, some energy is liberated, which is utilized in the construction of a bond between ADP and IP to form ATP. So, the energy utilized for synthesis of ATP is oxidation energy. So, by using the oxidation energy, if ATP are synthesized, which is commonly called oxidative phosphorylation. So, after seeing this, this is the only event in entire aerobic respiration where oxygen is involved. So, it is also called O2 dependent process. So, O2 dependent process in aerobic respiration is the ETS and oxidative phosphorylation, and oxygen participation is seen in this step only. Though there are several events involved in aerobic respiration, but oxygen participation is the direct in the last and final. Even though oxygen is involved in the last process, but it plays an important role in complete oxidation of glucose. So, if O2 is not involved here, there is no agent to accept electrons and protons of glucose. So, terminal electron acceptor is oxygen. As long as the O2 is available, the NADHs are oxidized into NAD pluses, FADH2s are oxidized into FAD pluses, which are going to be again accepts hydrogens from glucose. So, continuous oxidation of glucose requires enough amount of NAD pluses and FAD pluses. That implies NAD plus and FAD plus should be regenerated continuously. For regeneration of these two things, hydrogen acceptor availability is important. So, O2 role, though it is evident in the last step, but it has an important role in the completion of aerobic respiration. So, let us see the another aspect. So, during this fourth and final step, as I said, the NADH plus H pluses, one form of reduced coenzymes, they need to be oxidized. So, during oxidation, each NADH loses 2 electrons and converts into NAD plus and also loses 2 protons. Similarly, the FADH2 also oxidizes and also releases 2 electrons and forms into FAD plus and protons. So, 2 electrons and 2 protons from each reduced coenzyme, and these electrons are going to be transferred through a complex of electron carriers, which are usually found in inner mitochondrial membrane. So, like these are ubiquinone and cytochrome B, cytochrome C1, cytochrome C and cytochrome A, and finally the cytochrome A3. So, these are iron containing organic compounds which act as an electron carriers, and they are undergo a series of electron acceptance and electron loss, that is reduction and oxidation. So, through a series of oxido-reduction reaction, the electrons of NADH or FADH2 finally transfer to an oxygen atom. And this oxygen atom, after taking those electrons which are passed through the chain of electron carriers, reacts with protons, they are taken from matrix and converts into water. So, water is formed due to the reduction of oxygen, and reduction of oxygen is because of oxidation of reduced coenzyme, and electrons transfer is always takes place in accordance to their redox potential values. It is a common value, redox potential values. Every chemical has an ability to gain or lose the electrons. The ability of gaining and losing electrons by a chemical substance called redox potential. So, electron travel is always strictly according to redox potential scale. So, from NADH2 to oxygen or FADH2 to oxygen, electron travel via a chain of electron carriers occur as per the redox potential gradient. And electrons are definitely with an energy, while transferring from one component to another component, they lose some energy. So, in a downhill movement from high energy status to the low energy status, electrons are going to be lose energy, and that energy formed during the downhill of electron movement is utilized for the production of ATP. So, energy released during the electron transfer is utilized for the production of ATP. Hence, in this process, there is an electron transport from reduced coenzyme to the final electron acceptor, coupled with synthesis of ATP. That is why this event is known as ETC or ETS, Electron Transport System, and oxidative phosphorylation. The energy liberated during series of oxidative and reduction reactions being utilized for synthesis of ATP. So, two events occur one after another. So, the water that is formed in this reaction is called metabolic water. So, oxygen atom is a terminal electron acceptor, and this is a chain of electron carriers, electron transport chain, otherwise respiratory chain. So, in respiratory chain present in inner mitochondrial membrane comprises different types of electron carriers, which involves the transfer of electrons, where the electron carriers undergoes a series of reductions and oxidation. Finally, the electrons of low energy accepted by oxygen and reacts with protons, converts into water, and ATP is formed. So, regarding the electron transport chain and the oxidative phosphorylation, take this magnified portion of inner mitochondrial membrane. Since the inner mitochondrial membrane is very much equipped with the entire system required for completion of that so-called fourth and final step. And you know that inner mitochondrial membrane is a lipoproteinaceous membrane. There are two lipid layers in a uniform manner, and there are several proteins. So, these proteins are now going to be formed a multi-enzyme complexes, which are involved in completion of this final step. That is, so altogether for completion of this step, the electron transport chain and oxidative phosphorylation event requires the participation of some complexes. They are named as complex 1, complex 2, complex 3, complex 4, and complex 5. So, this fourth step requires 5 specific complexes. They are named as a complex 1, 2, 3, 4, and 5, and they are present in the inner mitochondrial membrane. Along with these complexes, for completion of this fourth step, they require 2 important molecules which are served as electron carriers. So, electron carriers like ubiquinone, UQ means ubiquinone, and cytochrome C. So, cytochrome C, ubiquinone are the 2 electron carriers, along with the complex 1, complex 2, complex 3, 4, and 5 involves in the finishing of last step. So, complex 1 composed of several components like FMN, and it has iron sulfur centers, FMN, and few iron sulfur centers. Complex 2 comprises FAD and iron sulfur components. Complex 3 has cytochrome B. There are 2 types of cytochrome Bs, and an iron sulfur center, and also cytochrome C1. So, cytochrome B, iron sulfur centers, and cytochrome C are the integral parts of complex 3 of ETC. Then, in a complex 4, there are 2 units of copper centers and cytochrome A and cytochrome A3. Cytochrome A and cytochrome A3 are the components of complex 4, and the complex 5 has two parts. One is F0 and another one is F1. So, F0, F1 parts of complex 5, and copper units 2 units, and cytochrome A, A3 are the components of complex 4. Cytochrome B, cytochrome C1 along with iron sulfur centers are the components of complex 3, and FAD and iron sulfur centers for complex 2, FMN and iron sulfur centers for complex 1. And moreover, these complexes are also maintain some enzymes. So, they have catalytic role. Complex 1 is chemical or catalytic name or enzymatic name is called NADH, ubiquinone NADH oxidoreductase, ubiquinone NADH oxidoreductase, and enzyme name of FAD is succinic dehydrogenase. So, these are important combination. The enzyme name of complex 1 of ETS, ubiquinone NADH oxidoreductase, and complex 2 enzymatic name is succinic dehydrogenase. For complex 3, the cytochrome C reductase. Cytochrome C reductase is the enzyme name of complex 3, and the cytochrome C oxidase is enzyme name of complex 4. Cytochrome C oxidase is enzyme name of complex 4. Like that, the F1 particle of this complex 5 is called ATP synthase. The enzyme name is ATP synthase. So, 5 different complexes with 5 different enzymes along with 2 electron carriers involved in the process of last and final event. So, let us take these functions. So, first of all, the reduced coenzyme which are formed in mitochondria. I am taking this mitochondrial NADH. Since in respiration, NADHs are formed in mitochondria during link reaction and Krebs cycle, and NADHs are also formed in cytosol during glycolysis. So, those are called cytosol NADHs. So, mitochondrial NADHs and cytosol NADH. So, whatever the location, they should be undergo oxidation. So, first of all, let us take NAD, mitochondrial NADH. So, it is oxidized by losing the electrons, 2 electrons and also 2 protons, and converts into NAD plus. So, these electrons from NADH plus H plus transfer to FMN, moves to iron sulfur component, and finally transfer to ubiquinone. So, during the oxidation of mitochondrial NADH, the electrons are transferred first to FMN part of complex 1, then iron sulfur center of complex 1, finally transferred to ubiquinone. In meantime, the protons are transported from matrix to perimitochondrial space. This is the space between perimitochondrial space. Is a place between outer and inner mitochondrial membrane. So, it is also called outer space or intermembrane space. So, protons which are released during oxidation of NADH are shunted into the intermembrane space. So, the protons are shunted or pumped into intermembrane space by using the energy liberated during electron transfer. So, the electron transfer energy is coupled with the proton translocation. So, proton translocation is a process where energy is utilized, that is during electron transfer. So, there is a coupling downhill of electron transfer coupled with uphill of proton translocation. So, there is an exergonic process. Energy is released, which is utilized for proton pump. So, two protons are added to the intermembrane space for pair of electron transfer. And ubiquinone, by taking the electrons and also protons, converts into ubiquinol, UQH2. So, ubiquinone is now converted into UQH2 after taking the electrons and protons. So, UQH2 is a mobile carrier, is a mobile electron carrier, also hydrogen carrier, and it transfers the electrons and protons to complex 3, where it oxidized into ubiquinone. So, UQ is reduced into ubiquinol by accepting electrons and protons, and ubiquinol, it is called UQH2, is again oxidized by donating electrons to cytochrome B of complex 3. So, the electrons from cytochrome B transfer to iron sulfur, and a final electron transfer to cytochrome C1 of complex 3. And finally, these electrons are transferred to cytochrome C. So, ubiquinol is going to be oxidized by donating electrons to complex 3. So, complex 3 is now reduced. So, oxidation of UQH2 leads to reduction of complex 3, and during which the electron transfer coupled with pumping of two protons. So, a pair of electrons transfer from ubiquinol to cytochrome B, iron sulfur, and cytochrome C1 of complex 3. Two protons are released, and these electrons are taken by cytochrome C. So, now cytochrome C is reduced. The reduced cytochrome C, which is located on the outer surface of inner membrane, which is a mobile one just like UQ, but UQ is an embedded protein. Cytochrome C is a surface or peripheral electron carrier, and it transfers the electrons to complex 4 and become oxidized. So, it is reduced by complex 3 and it is oxidized by complex 4. As I said earlier, there is a sequence of reductions and oxidation, and cytochrome C is capable of move freely between complex 3 and complex 4. So, it may be called shuttler. It shuttles between complex 3 and 4, involves in electron transfer. So, the electrons that are transferred from cytochrome C to complex 4, they are carried through various components of complex, first copper, then cytochrome A and cytochrome A3. Finally, the electrons of NADH of mitochondria transferred to oxygen atom by the last electron carrier complex, that is 4, with a compound cytochrome A3. So, the less energetic electrons from complex 4 accepted by half oxygen atom or half oxygen molecule and 2 protons from matrix together form one molecule of H2O. So, there is a journey of pair of electrons from mitochondrial NADH via complex 1, ubiquinone, complex 3, cytochrome C, complex 4, finally the ultimate electron acceptor oxygen, which reacts with the 2 protons and forms water. During this transfer of electron, complex 4 utilizes the electron energy and pumps 2 more protons. So, protons are slowly accumulating in intermembrane space as the electrons are moving from NADH to the oxygen. So, this completes electron journey from NADH to oxygen. But during this electron journey, according to redox potential, there occur pumping of protons into the membrane or into the intermembrane space. As a result of this electron transfer coupled with proton translocation, more and more positive charge increased on one side of the membrane and negative charges develop on the other side of the membrane. That means, the pH of the matrix going to be increased and the pH of the intermembrane space decrease. So, there is a pH gradient. So, electron transfer coupled with proton translocation results proton gradient across the membrane, and that proton gradient develops into a force called proton motive force. So, more and more protons are accumulated here, less protons are seen in the mitochondrial matrix. So, as per the thermodynamic principle, the region with more concentrated ions, where from there the ions supposed to move to their lower concentration. So, here in intermembrane space there are more protons, relatively less protons in mitochondrial matrix. So, these protons to be transferred from the region of their higher concentration to the region of lower concentration. But unfortunately, intermembrane space and mitochondrial membrane, mitochondrial matrix separated by inner mitochondrial membrane, which is a selective permeable. It never allows the free movement of protons, but it allows the movement of proton from higher concentrated to lower concentrated region via a complex, that is complex 5. So, the return flow of protons from mitochondrial perimitochondrial space to the matrix requires a complex, that is complex 5. So, these protons whatever accumulated in the intermembrane space according to their concentration gradient, they now moves through the complex 5. So, complex 5 has channels like F0. So, through this channel protons are moved and finally reaches F1 particle and released into matrix. And protons are also maintain some energy. That energy is released during their exit. So, that energy liberated during the proton transfer is utilized for the formation of a bond between ADP and IP. So, there forms ATP. So, proton energy is utilized, not protons. So, proton energy is utilized for synthesis of ATP. So, this proton translocation occurs because of gradient, and proton gradient is established because of electron transfer, and electron transfer occurs during oxidation. So, there is a combination. The electron transfer coupled with the proton translocation, which creates the proton gradient across the membrane, that finally develops a force called proton motive force, which drives the synthesis of ATP. So, this story explained clearly by Peter Mitchell in his theory called chemiosmotic theory. So, chemiosmotic theory proposed by Peter Mitchell. So, in mitochondria as well as in chloroplast, ATP synthesis occurs in accordance to Peter Mitchell's chemiosmotic theory, which says the electron transfer through membranes coupled with proton translocation, which creates a proton gradient that develops a proton motive force. That proton motive force finally utilized in the synthesis of ATP. So, oxidative phosphorylation in mitochondria during respiration, photophosphorylation in chloroplast during photosynthesis strictly occurs in accordance with chemiosmotic theory. So, one mitochondrial NADH transfer involves 6 protons accumulation, and these 6 protons return flow, say 6 protons return flow, there forms ATP. As per the chemical characters, if 2 protons are transferred through F0 and F1 particle, according to their gradient, the energy is sufficient to form 1 ATP from ADP and so as per the chemical energy calculations, every 2 protons travel through complex 5, 1 ATP is synthesized. So, here for mitochondrial NAD oxidation, how many protons are accumulated? 6. So, 6 protons travel yields 3 ATP. So, through 5th complex, if 6 protons are traveled, there are 3 ATP. So, 1 molecule of mitochondrial NADH oxidation results formation of 1 water molecule, synthesis of 3 ATP and utilization of half O2. So, like that mitochondrial NADHs are oxidized. So, another reduced coenzyme, that is FADH2. During the conversion of succinic acid into malic acid, hydrogens are removed, which are taken by FADF complex 2, converts into FADH2, and FADH2 is oxidized by releasing its electrons to ubiquinone and protons and converts into FAD. So, second complex is involved in the oxidation of succinic acid to malic acid. So, without participation of complex 1, complex 2 carries the transfers electrons of succinic acid straight away to ubiquinone. So, bypassing the complex 1, the FADH2 is oxidized by donating the electrons to ubiquinone, from there to complex 3, from there to complex 4, finally to oxygen. So, if this is the line, you will see only 4 protons are accumulated. So, oxidation of a mitochondrial FADH2 through ETS results 4 proton accumulation. So, 4 proton accumulation transfer results 2 ATP. Since 2 protons forms 1 ATP, so there are 4 protons which forms 2 ATP, utilizes 1 oxygen atom and forms 1 water molecule. So, like that, second complex involves in the oxidation of FADH2. First complex involves oxidation of mitochondrial NAD. Apart from these two, there is a cytosol NADH is there. So, it is also going to be oxidized after entering into the mitochondria and transfers its electrons and protons to ubiquinone. So, just like oxidation of FADH2, cytosol NADH also oxidized. So, here from cytosol NADH, electrons and protons straight away transfer to ubiquinone, which converts into ubiquinol, then electrons to complex 3, protons into mitochondrial matrix, mitochondrial inner membrane space, then cytochrome C, complex 4 and oxygen. So, with the completion of cytosol NADH oxidation, again 4 protons are accumulated, 1 oxygen atom is utilized, 1 water molecule is formed and 2 ATP are synthesized. So, cytosol NADH oxidation starts from ubiquinone, follows complex 3, cytochrome C, 4 and oxygen. But mitochondrial NADH oxidation starts with complex 1, ubiquinol, complex 3, cytochrome C, 4 and oxygen. So, different types of reduced coenzymes oxidation yields different amount of ATP. So, a brief summary of this oxidation. So, oxidation of 1 mitochondrial NADH, where a pair of electrons pass through electron transport chain, finally reach to oxygen to form water, during which 4, 2 plus 2 plus 2, 6 protons are accumulated here, which are written back to the matrix via complex 5, during which the 6 proton energy utilized in the synthesis of 3 ATP. So, correct sequence of electron travel during oxidation of mitochondrial NADH starts with complex 1, ubiquinone, complex 3, cytochrome C and complex 4. Finally, terminal electron acceptor in respiration is O2, forms water. So, there are nearly 6 NADHs are formed in TCA cycle and 2 NADHs formed in link reaction. So, altogether 8 mitochondrial NADHs are formed. So, through ETS, these 8 mitochondrial NADHs oxidize and forms 24 ATP. So, 1 mitochondrial NADH results 3 ATP. There are 8 mitochondrial NADHs altogether formed during aerobic respiration in this split. Finally, 24 ATP. So, next one is oxidation of FADH2. 1 molecule of FADH2 on oxidation, pair of electrons are released, passing through ETS, finally accepted by oxygen and 1 water, during which 4 protons are accumulated and these 4 protons return through complex 5 to form 2 ATP. So, correct sequence of electron travel during FADH2 oxidation, complex 2, not complex 1. From there, ubiquinone. From there, complex 3, C, and finally 4, lastly, oxygen. So, in this case, there are 2 FADH2s are formed during the TCA cycle. So, 1 FADH2 oxidation results 2 ATP. So, altogether through ETS or ET last oxidation, 2 into 2, 4 ATP molecules are formed on oxidation of 2 molecules of FADH2. So, there is one more thing is there, that is mitochondrial NADH oxidation. Apart from that, cytosol NADH. So, let us see the cytosol NADH oxidation. So, one cytosol NADH, that is the NADH formed during EMP pathway, also take part in oxidation. So, during its oxidation, the pair of electrons from cytosol NADH pass through electron transport chain, reach to oxygen and forms one molecule of water, during which there are 4 protons are accumulated in the intermembrane space, just like oxidation of FADH2. Those 4 protons are written back into the matrix via complex 5, which yields 2 ATP. So, 1 cytosol NADH oxidation yields 2 ATP. So, how many cytosol NADHs are formed during aerobic respiration? Generally 2. So, oxidation of 2 cytosol NADHs through ETS forms 2 into 2, 4 ATP. And a correct sequence of cytosol NADH oxidation from cytosol NADH to ubiquinone, complex 3, cytochrome C, 4 and oxygen. So, mitochondrial NADH oxidation yields 3 ATP, FADH2 oxidation yields 2 ATP, cytosol NADH oxidation also yields 2 ATP. So, different types of reduced coenzymes on oxidation produce different number of ATPs. So, after seeing all these things, so total ATP generated through oxidative phosphorylation, it comes like this. There is an oxidation of 8 mitochondrial NADH yields 24, right? And oxidation of 2 FADH2s yields 4, and oxidation of 2 cytosol NADH also yields 4 ATP. Summing all this, so what comes? 24 plus 4, 28, 28 plus 4, 32. So, altogether 32 ATP molecules are generated on complete oxidation of all reduced coenzymes. And what are the number of reduced coenzymes? 8 plus 2 plus 2. That means 12 reduced coenzymes are formed during the aerobic respiration. All these 12 reduced coenzymes in this form, 8 mitochondrial NADHs, 2 cytosol NADHs and 2 FADH2s participate in electron transport system and converted and transfer their electrons to oxygen, finally forms 32. So, this is the number of ATP molecules formed through oxidative phosphorylation, 32 ATP. So, the total summary of aerobic respiration, which includes or which provides the information regarding the steps their occurrences, various reactants, products, and finally the result, ATP. So, because respiration is a process through which the cell gets energy in the form of ATP from a substrate. So, let us take one molecule of glucose, how many ATPs are totally produced and how many ATPs are utilized, and finally net amount of ATP. So, you can go through these things and finally you will get 36 ATP, of which 32 are formed as a result of oxidative phosphorylation and 4 are formed as a result of SLPs. So, through SLP, the 4 ATP, through oxidative phosphorylation 32, so altogether 36. So, one molecule of glucose oxidation results 36 ATP and 6 CO2 molecules are released. So, it may be useful to you. So, next type of respiration is aerobic respiration. So, aerobic respiration is another type where oxygen is not utilized, but same type of respiratory substrate glucose is going to be participated. In the absence of oxygen, glucose is going to be partially oxidized and there is only limited amount of energy is liberated. CO2 is liberated, but ATP number is restricted to only 2 per glucose molecule. So, during this process, there is a formation of some organic molecules like ethyl alcohol in this context, sometimes lactic acid. So, based on this last formed product, the aerobic respiration is named alcoholic fermentation or followed by lactic acid fermentation, or anaerobic respiration may be followed by butyric acid fermentation. For example, alcohol fermentation occurs in some examples like yeast. So, during that respiration by yeast in the absence of oxygen, glucose converted into 2 molecules of pyruvic acid through 10 enzymatic reactions in cytoplasm, where 2 ATP are utilized, 4 are formed. So, net is 2 and 2 molecules of NAD is reduced into 2 molecules of NADH. So, what is it? EMP pathway as it is. But after that, as oxygen is not available, so pyruvic acid converts into acetaldehyde and finally converts into ethyl alcohol. So, the fate of pyruvic acid formed after EMP depends on availability of oxygen. Here oxygen is not available. So, instead of undergoing complete oxidation and decarboxylation, pyruvic acid undergoes decarboxylation and then reduction. So, this is the process reduction and this is decarboxylation. So, carbon dioxide is released, acetaldehyde is formed, which is a 2 carbon compound. The acetaldehyde is reduced into ethyl alcohol, where hydrogen is donated by NADH plus H plus formed during glycolysis. So, there is no possibility of oxidation of cytosol NADH to get extra ATP. Hence, in anaerobic respiration followed by fermentation, very less amount of ATP is generated, 2 molecules of CO2 and 2 molecules of ethyl alcohol. So, see the product of EMP pathway is called pyruvic acid and it is very much known as a key product of EMP pathway, whose conversions further depends on availability of oxygen. It was proved that in the presence of oxygen, pyruvic acid undergoes some reactions. In absence of oxygen, pyruvic acid converts into some other. So, the fate of pyruvic acid depends on availability of oxygen. In absence of oxygen, say anaerobic conditions, there are changes. The pyruvic acid in absence of oxygen may be reduced into lactic acid straight away. So, lactic acid is formed from pyruvic acid when oxygen is not supplied. And this is a very commonly occur in human skeletal muscle cells whenever the body is experiencing severe stress. That is a continuous uncontrolled exercises results non-availability of oxygen to the EMP pathway product. Hence, the pyruvic acid in human skeletal muscle cell converts into lactic acid by means of NADH involvement. And then in yeast, there is a Saccharomyces, a unicellular fungus. In absence of oxygen, in yeast cells, the key product of glycolysis first undergoes decarboxylation, then forms into acetaldehyde, which later undergoes reduction, not oxidation, reduction converts into ethyl alcohol. So, COT is formed, ethyl alcohol is formed, but no ATP in this case. Lactic acid formation does not yield ATP. Ethyl alcohol formation does not yield ATP. So, under anaerobic condition, whatever the energy present in glucose is trapped only in 2 ATP. So, anaerobic cell supposed to be satisfied with only 2 ATP. But coming to the presence of oxygen, in the presence of oxygen, the pyruvic acid moves into mitochondria, converts into acetyl coenzyme A through link reaction, and then participate in TCA cycle, where it is completely oxidized and decarboxylated into water and carbon dioxide, a good amount of NADHs, good amount of FAD is formed. All these NADH, FAD formed in mitochondria undergoes oxidation through ETS. So, there are 32 ATP. So, 32 ATP are generated in mitochondria, 2 ATP are generated in glycolysis, and 2 more ATP generated in mitochondria through TCA cycle. So, altogether from one glucose molecule, if oxygen is there, the pyruvic acid is finally decarboxylated and oxidized, yields 36 ATP. So, fate of pyruvic acid depends on availability of oxygen. So, respiration, that is of course aerobic respiration is also called amphibolic pathway because of its characteristic nature. So, amphi means double. So, the respiration is going to be show two different types of changes. It acts as generally it works as a catabolic process already studied, where glucose is degraded into carbon dioxide and water and energy. So, glucose, fructose 1,6-bisphosphate broken into PGAL and DHAP, later pyruvic acid, acetyl coenzyme A and TCA cycle. And also if proteins are involved, proteins are acts as a respiratory substrate, but they cannot directly enter into respiration. They degraded into first amino acids, which loses ammonia, deamination, and found converts into acids like either pyruvic acid or alpha-keto glutaric acid. So, from proteins via amino acids and keto acid, the respiration starts. In fats, if at all acts as a respiratory substrate, they cannot directly respire. So, because of that complexity, they first broken down into fatty acids and a glycerol, and a glycerol converts into PGAL, take part in respiration. Fatty acids converts into acetyl coenzyme A, take part in respiration. So, in all these cases, whether glucose or fats or protein, there is a catabolism is there. But sometimes The acetyl coenzyme formed in respiration may leave the pathway and utilized in the synthesis of fatty acids, in turn fats. So, there is a construction of substances from a respiratory intermediate. And alpha-ketoglutarate acid, after taking ammonia, converts into glutamic acid, which take part in proteins. So, the another line is construction. So, both destructive process, the broken lines, and constructive process, the strong lines, occurs during respiration. Hence, the scientists named respiration as amphibolic pathway. There is a catabolism and anabolism.